One of the most challenging problems in the field of medical science today is predicting the drugs that will work for specific cancer patients. Currently, research shows that pathway-targeted drug therapies vary greatly in effectiveness depending on the cancer cell type. Thus, no prediction of the efficacy of a specific drug can be made. The hypothesis for this phenomenon is that various cancer cell types have different activated pathways, and drugs targeted to a specific pathway will only be effective if that pathway is indeed upregulated. An upregulated pathway is defined as having a higher activity of protein and transcript levels in the cell. The objective of my project was to develop a software tool to test my hypothesis and identify and analyze upregulated pathways in malignant cells through a unique heuristic data-driven approach by analyzing the interaction between various data sets. Essentially, I wanted to be able to identify upregulated pathways across varying cancer cell lines and then be able to see if there was a correlation between upregulation and positive drug response. Specifically focusing on non-small cell lung cancer, 3 phases of this project were carried out to validate the hypothesis. Phase 1 was data discovery and analysis, where I essentially collected and integrated and developed a software tool to analyze a data cube consisting of 3 different types of data: proteomic, transcriptomic, and drug sensitivity data. Phase 2 was statistical inference, where I essentially built various statistical analysis tools such as probability density, bell curves, scatter plots, and paired p-value calculators to be able, using PHP scripts and math API libraries, to first of all be able to determine the statistical difference between pathways and then determine if that difference was actually upregulation. Phase 3 was dynamic visualization. One of the biggest problems in the field of biology today is being able to model the dynamic reactions that occur in a cell. Currently, the only software programs that are available are create static images of these, such as Cytoscape. However, these software programs don't show the dynamism of the processes that occur in cells. So to address this problem, I essentially created a software program that visualizes dynamic models of biological networks to simulate cell interactions with data analysis. For our results, I was able to effectively validate the hypothesis with 94% of pathways showing that they were upregulated and had positive drug response. Moreover, I was able to view various biological data such as transcriptomic, proteomic, and drug data as a system and effectively determine the correlation between those data sets. Finally, being able to create a system to visualize dynamic models with the integration of biomolecular signaling networks. In terms of conclusion, the end product of this project was essentially a user-friendly data analysis and visualization software. The bigger picture is when a patient gets diagnosed with cancer, the first thing they do is get their tumor resected. Clinical researchers currently are working on making a tumor profile of that specific tumor, and that data, such as protein data and mRNA data, can be uploaded to my software, and my software can create a complete pathway analysis for a specific patient based on the patient's tumor analysis. Now, this software can successfully help clinicians narrow down the number of drugs